Okay, we're back, continuing on with chapter 9. I think we left off here talking about monohybrid crosses. And now we're going to talk about a dihybrid cross. What would result from a dihybrid cross, a cross between two organisms that are each heterozygous for two characters. So we're following two characters at the same time. And what men wanted to know was, are these characters somehow linked? The factors that control these characters, are they somehow linked? And do they travel together? Or the way when they're segregated? Or are they segregated independent of each other? They don't really have anything to do with each other. That would be called independent assortment. If they're somehow linked and they go together, that's dependent assortment. If they're the segregation is each independent event, that's independent assortment. So that's what he wanted to find out. So this is the question. So we cross the F1 plants to get with each other, and if the genes for two characters were inherited together, then the F1 hybrids would produce only the same two types of gametes that they received from their parents. And the F2 generation would show this 3-to-1 phenotypic ratio between the two phenotypes, right? If they join together, if we had dependent assortment. If, however, the two seed characters were sorted independently, then the F1 generation would produce 4 different gametes, gamete genotypes in equal quantities, and the F2 generation would have 9 genotypes producing 4 different phenotypes in a ratio of 9:3:1. And you gotta just remember this 9:3:1. That's 16. Because we're going to do the monohybrid cross. Hybrid cross was 2x2. This is going to be a 4x4. It's 16 different genotypes. So that's where 9:3:1 comes in. That's 16. So 9/16 we're going to be one type of phenotype. 3/16 one type. 3/16 one type. 1/16. Which is all recessives. So it really works out to be 56% for one phenotype, about 18.75% or 19% for the 3/16, and 1/16 is 6%. So if you get if you have a dihybrid cross, you get this 9:1 ratio, you say independent assortment. If you don't get that ratio, it's not an independent assortment. So the Punnett square in figure 9.8b also reveals that dihybrid crosses are equivalent to 2 monohybrid crosses occurring simultaneously. It's going to look a little more complicated, and it is a little more complicated, and there's one extra step you've got to do a dihybrid cross. Let's first look at, so it's hypothesis dependent assortment to the left, and then and that would produce only, so we're looking at yellow seeds versus green seeds, and we're looking at smooth seed versus wrinkled seed. So it's dependent assortment. So now in a dihybrid cross, a genotype is 4 letters, right? Because it's 2 loci. So this is one the R locus and one the Y locus. So this PJ, if it's dependent assortment, so we're starting with the parentals, is all both loci are homozygous dominant. Big R, big Y, big Y. So that is yellow smooth. Yellow dominant over green, smooth dominant over wrinkled. And then you cross that with a small r, small y, homozygous recessive at both loci. And then you got to select the gamete combinations. This is the extra step in a dihybrid cross. Because now you put 2 letters in the gametes, not just one from each locus. So it's got to be an R in there, and it's got to be a Y in there. Now, if it's dependent assortment, and a big R and a big Y always go together, then there's only 2 types of gametes that can be made. Big R, big Y, small r, small y. And the F1 generation would be a double heterozygote. Big R, small r, big Y, small y. And then you'd have this kind of a predicted result right here. 3-to-1 between the two phenotypes, yellow smooth and green wrinkled. If it's independent assortment, then you're going to be making what are called recombinant gametes. And that is, you won't just make big R, big Y, and small r, y. You also make small r, big Y, and big R, small y. Those are called recombinant gametes. And you're going to have 16 genotypes. Not all of them are unique. And you're going to have the parental phenotypes, round yellow and wrinkled green, but you're going to have recombinant phenotypes, round green and wrinkled yellow. All right, if they're independent assorting. So that's the so here's the parental cross. That's a predicted result for dependent assortment, and it wasn't seen. What he got was these 4 different phenotypes rather than just 2. So he had concluded these 2 traits were these 2 characters were independently assorted, right? So Benno's dihybrid cross supported a hypothesis that each pair of alleles segregates independent of the other pairs during gamete formation. Thus, the inheritance of one character has no effect on the inheritance of another. This is called Mendel's law of independent assortment. And he thought, and remember, he tested 7 characters, and he did the dihybrid cross between 2 of all of them. And every time he did a dihybrid cross, he got independent assortment. So he thought it was a universal law for all characters, genetic characters. It's not. And I'm going to explain in a little bit more what independent assortment really means. And it was just by sort of like luck that all 7 of his characters they tested independently sorted. And it has to do with what chromosome they're on. We're on same chromosome or different chromosomes. I'll explain that later. Okay, here's independent assortment seen in the inheritance of 2 characters in Labrador retrievers. So possible phenotypes of Labrador retrievers. Now, there's 2 characteristics, seeing and blind, black coat, chocolate coat. So there's big B for black coat, which is dominant over small b, chocolate coat. Seeing, big N, is dominant over blindness, small n. So what would be the genotype of a dog here with a black coat and normal vision? Gotta have a big B because it's black and something else. And that could be a big B or a small b, wouldn't matter because big B is dominant. And it has to have a big N. And it doesn't matter what the other one. Remember, there's 4 letters in a dihybrid genotype. So it's big N and something else, big N or small n, doesn't matter. Now, black coat blind, black coat means it's got to have at least 1 big B, right? But since it's blind, the other locus has to have 2 small letters. So big B something, small n, small n. Then chocolate coat, normal vision. Chocolate coat can have 2 small letters. That's recessive, so small b, small b. But it has normal vision, so it's got to have a big N and something else. And then finally, the chocolate coat blind, that's all small letters. Small b, small n, small n. And when you make these dogs together, you get 9 black coat normal vision, 3 black coat blind, 3 chocolate coat normal, and 1 chocolate coat blind. 9, 3, 3, 1. So what is the ratio of a black coat to chocolate coat, or normal vision to blindness? Let's just make believe. Let's just make believe it's doing a monohybrid cross. All right. So let's look at each one in a monohybrid cross. Let's just look at black animals versus the chocolate animals. All right. How many black? You have 9 black and 3. That's 12. We got 3 chocolate and 1 chocolate. That's 4. That's 12 to 4. What ratio is that? 3 to 1 for the coat. Same thing with blindness. How many blind animals do we have? Or normal. Okay, first one, how many normal? Got 9 normal here, 3 normal here, 12 out of 16 are normal vision. How many blind animals? 3 here, 1 there, that's 4. 12 to 4, same thing. It's 3 to 1. So it's like 2 nested monohybrid crosses. Whoop. So there's the 4 phenotypes, and those are the possible genotypes for each one of those phenotypes. And you're mating 2 double heterozygotes, big B, small b, big N, small n, times big B, small b, big N, small. What are those? What's the phenotype of both of those parents? Black, normal vision, because they have a big B and a big N. Okay, now, test closer took back to independence. So I am going to pause this here and go on the board and try to explain really what the nature of independent assortment is before we go on to this other stuff. So I'm going to pause this moment. I'll be right back. Okay, we're back. I'm going to try to use the board here to explain independent assortment and how it has to do with whether the alleles are on the same chromosome or different chromosomes. Here's an individual that's a double heterozygote. Big B, small b, big N, small n. And the 2 loci, I'm using the dog example here. So let's say the B locus and the N locus are on different chromosomes. And this is, you should recognize this as metaphase 1 of meiosis, where the homologous chromosomes line up, okay? So what if they line up like this? Then in anaphase 1, these 2 will go over here, and these 2 will go over here. So what kind of combination will we have over here? Big B, big N, big B, big N. And over here, what allele combination will we have in the gametes? Small b, small n, small b, small n. See? Okay, that's how we can get what are called the parental combinations. But what if these homologous chromosomes lined up differently? For example, what if the one with the big N lined up over here, and the one with the small n lined up over here? So now this is small n, big N, big N. Well, this we could have switched. Big B could be over here. See, the way these line up in metaphase 1 is independent of each other. So they are sorting independently. If they line up like this, then what kind of gametes do you have over here? Big B, small n. And that would be a recombinant, called a recombinant combination, because that's not going to be a situation found in parents. It's going to show up as one of those recombinant phenotypes, like the black blind or chocolate normal. And then over here, you end up with the other recombinant combination, small b, big N. And that's how, and all four of those gamete combinations are equally probable. So this will give you the 9-3-3-1 ratio. And you say these are independently assorted, which means that they're on different chromosomes. Well, probably on different chromosomes, because I'm going to give you an exception to that. You can't really say for sure whether, well, first let me give you a situation if they're on the same chromosome, dependent assortment. Let's put them on the same chromosome. All right, let me erase this. Put them on the same chromosome. So what happens when they, in a phase 1, we got big B, big N, and small b, small n over here, right? Well, what if they line up differently, like this? Well, it won't make any difference. If they line up so that the two small letters on this side, we'll have this one over here, and two big letters over here. So see, it doesn't make any difference which way they line up because they're on the same chromosome. So the big B and big N go together, and the small b and small n go together. So this would not be independent assortment. You wouldn't get the 9-3-3-1. You'd get something really different. You'd get something like, well, you'd probably get 9. And then over here for the double dominant, and you'd probably get like, let's make it, no, 5, and that's 14, and then 15, 16. You get something like 9-1-1-5 or something. Be way off in a 9-3-3-1. Therefore, you'd say these are not independently assorted. But you still get four different phenotypes. You still get their recombinant phenotypes, even if they're on the same chromosome. How can that be possible? Well, what makes it possible is crossing over. What if there were a crossover event between these two non-sister chromatids? Crossing over. So the small b goes over here, and the big B goes over here. Now, when they separate, you're going to get small b, small n, but you're also going to get big B, small n. And you're going to get the big B, big N over here, but you're also going to get big N, small b. So you can get the recombinant gamete combinations, even if they're on the same chromosome, if there's a crossover event between them. But here's the thing, and crossing over always happens. The thing is, the closer they are together on the same chromosome, the less likely a crossover event is going to occur. So the rarer the recombinant phenotypes. And the farther apart they are in the same chromosome, the more likely you will be to get the recombinant phenotypes. And as a matter of fact, if they're far enough apart on the same chromosome, they will independently assort, because if you have 50% chance, if there's a 50% chance of getting a crossover event between them, they will independently assort, even though they're on the same chromosome. That's why if they do independently assort, you can say they are probably on different chromosomes. Either that or very far apart on the same chromosome. But if they don't independently assort, you can definitely say they are on the same chromosome. And these are called linked genes if they're on the same chromosome. And as a matter of fact, that's a situation with Mendel. Two of his characteristics were on the same chromosome, but they were so far apart that there was a 50% chance of a crossing over, and therefore they independently assort. Because he didn't know anything about that, about chromosomes or crossing over, or meiosis, or anything like that. So he thought, as I said, he put down a law of independent assortment. He thought that all characters independently assorted. Well, they don't. They don't if they're close together on the same chromosome. Okay. So let me pause again and go back to screen sharing, back to the PowerPoint. Let's pause. Okay, let's get back to PowerPoint now, and let's talk about test cross. Now here's the thing. Let's say you have an individual that's exhibiting the dominant trait of a particular character, all right? And you want to know whether that, now you can't know for sure the genotype of an individual that is expressing the dominant trait of a character, because both homozygous dominant and heterozygous has the same phenotype, the dominant phenotype. So let's say you, for some reason, you want to know whether that individual is homozygous dominant or heterozygous. Maybe you have some financial interest in this, and so how are you going to find out? You can't dissect them and see whether they're homozygous dominant or heterozygous. What you do is do what's called a test cross. You mate that individual with an individual that you know what their genotype is just by looking at it. And who is that? The homozygous recessive. The only individual that can exhibit the recessive trait is the homozygous recessive individual. So you mate that unknown individual with the homozygous recessive, and you look at the offspring. Okay, if you made a chocolate lab with a black lab of unknown genotype, so black lab is unknown genotype because that's the dominant, right? And you end up with a litter of 3 black puppies. Do you know that for certain the genotype of a black parent? No, you don't. But it's likely to be homozygous dominant, because if you if the black animal were heterozygous and you made it with the homozygous recessive, there'd be a 50% chance of having the recessive phenotype. Maybe I'll do that on the board here. So you do a Punnett square. Small b, big B, small b, right? Big B, small b, big B, small b, small b, small b, small b, small b. So it's a 50% chance of getting the recessive phenotype. If this was big B over here, like to the left on the PowerPoint, then there'll be no chance whatsoever of getting a chocolate lab. They would all be black because they'd all be heterozygous, you see. So if you get even one, if you do a test cross and you even get one offspring that has the recessive phenotype, you know for certain that that unknown individual is heterozygous at that locus. If you get a big litter and they all are black, then then you could say with a good degree of certainty or good degree of confidence that the unknown individual is big B, because if you get a big, now 3 litter, that's a little small, but if they get a big litter and they're all black, it's likely that the unknown individual is big B, because there's a 50% chance of having a chocolate lab. If you get a big litter, you'd think that half of them would be chocolate, but they're all black. So you could say you can't be absolutely positive, but you can be kind of sure that. So you can't be absolutely certain that the individual is big B. But if you get, like I said, if you get even one individual with the recessive phenotype, then you are certain that the unknown individual is heterozygous. Okay. Okay. Mendel's strong background in mathematics helped him understand patterns of inheritance. You see, this, like I said before, he looked up the ratios. Nobody had done that before. It's such a simple thing, and we think, wow, of course, but nobody had done it. So he understood that genetic crosses obey the rules of probability. The same rules that apply when tossing coins, rolling dice, or drawing cards. Rule of multiplication states that the probability of a compound event is the product of the separate probabilities of the individual events. So you're flipping coins. It's just like making games, heads or tails. You got a 50% chance of having a head or a tail. So half of the time you have a head, half the time tail, half the time head, half the time tail. So what's the probability of getting two big B's? You multiply 1/2 times 1/2, 1/4. So it's 1/4 for each of these. So it's a 1/4% chance of big B, 1/4% chance of small b, and 1/2 and 1/2 50% probability for big B, small b, because there's two ways to get it. Here, this one is 1/4, and that one's 1/4. So you add those together to get 1/2, or 2/4. Okay, so geneticists who study people obviously cannot control the mating of the research participants. Instead, they must analyze the results of matings that have already occurred. And this is, family's history, genetic history, a pedigree, a family tree. And so Mendel's laws enable us to deduce the genotypes from most of the people in the pedigree. Here's a pedigree for freckles versus no freckles. So freckles is dominant, no freckles is recessive. So here's Aaron and here's Betty. Let's make believe you didn't know that they were heterozygous because they're exhibiting the dominant phenotype, freckles. So you don't know whether they're homozygous dominant or heterozygous. But you look at their offspring. You couldn't tell with Evelyn and Hal, but Fred and Gabe don't have freckles. They show the recessive phenotype. Oh, by the way, females are circles, males are squares in all of these pedigrees. Okay, and then if the individual in the pedigree shows the dominant phenotype, then you don't fill it in. If it shows the recessive phenotype, you fill it in, right? So Fred and Gabe have filled in squares, so they don't have any freckles. It's recessive. And so here's the thing. So then absolutely for positive sure that Fred and Gabe's parents, Aaron and Betty, are heterozygous. Why? Because Fred and Gabe have two small letters. They had to get a small letter from mom and a small letter from dad. So both parents have to have a small letter. If all four of those children had freckles, then you could say, well, one of those individuals, Aaron or Betty, is probably big F, and there's no possibility of getting a freckleless child. Okay. See what I mean? And also, if you see this, that two parents don't show a trait, and it shows up in the offspring, you also know it's recessive. It has to be recessive. Okay. Now here on this side, Cletus and Debbie. Now Cletus doesn't have any freckles, so Debbie does, and she's heterozygous. So we have Julia with small f, and then so then you could know that Debbie is heterozygous, and all the children are going to get a small f. So you know that Ina is going to be heterozygous. Why? Well, she has freckles, so she's going to have a big F. And her father is Cletus without freckles, so the only thing he can give is a small f. So you know that she is heterozygous. And there's going to be the same cross as Aaron and Betty. Lisa with either big F, or big F, small f. But Karen, you know, is small f, small f. So you can't always tell exactly what they are, like Evelyn over here, or Lisa, because they haven't made babies and looked at the babies. So they're either big F or big F, small f. But since Hal made a baby with Inna and they had Karen, then you know that Hal is heterozygous. See, so sometimes you can know for certain, other times you can't know if they didn't have any children and you can't look at the children. Okay, so there's the pedigree. There's pedigree analysis. And so if you have a particular genetic disease or some kind of condition in your family, you can go back and look and see who in the family had it and whether, and most genetic diseases are recessive, so they're hidden, and you could trace what is the probability, what's the chance that you are carrying this, that you're a carrier for. And that's what a carrier is. A carrier is a person or an organism that carries a recessive allele that is heterozygous for recessive allele, that could be a really dangerous recessive allele that causes a genetic disease. So it could just not be the lack of freckles. I mean, okay, a trait that is dominant does not imply that it's either normal or more common than a recessive phenotype. Instead, dominance means that a heterozygous genotype results in a dominant phenotype. Wild type traits, those most often seen more often in nature, are not necessarily specified by dominant alleles. They often are. You expect to see more dominant individuals with dominant traits rather than recessive traits, but not always. Wild type traits are the most common. And here's the thing. We're going to look at blood types in a bit, and that's a little more of a complicated situation. But we're going to learn that blood type A and B are dominant over blood type O. O is actually recessive. But if you look at the human population as a whole, A is most common, but O is second most common. Even though it's recessive, it's second most common. So, like I said, it's not always the case, but wild type traits are usually dominant, but not always. Okay, the genetic basis of many human characteristics, such as eye and hair color, are complex and poorly understood. Mendel's laws can be applied to the inheritance of human traits that are generally agreed upon to be determined by a single gene, such as freckles or widow's peak hairline. But we're going to see that we're going to get into later on in this chapter what are called non-Mendelian traits. These Mendelian traits, or characters, I should say, are those that exhibit the laws that Mendel figured out, and that is completely dominant, completely recessive. One gene, one characteristic, all right? And independent assortment. If they exhibit all of that, that's those are called Mendelian loci. But we're going to get into, but they found out, you know, after they in the early part of the last century, when they started, when they found Mendel's work and they started looking into genetics more, they found out that inheritance is a little more complicated. There's lots of different ways that traits are coded for in genes, and these are called non-Mendelian characteristics. And we're going to get into those later on in this chapter. Okay. So the human genetic orders listed in Table 9.1 are noted to be inherited as dominant or recessive traits. These disorders therefore show simple inheritance patterns like the one Mendel studied with pea plates. Like widow's peak or straight hairline, dominant trait, recessive trait. And all of these disorders, albinism, cystic fibrosis, phenylketonuria, sickle cell disease, and the worst of the worst, Tay-Sachs. And we are all tested at birth for a lot of these, particularly PKU. Moments after you're born, the physician will make a little cut in your heel take a few drops of blood and test you for all of these, for many of these genetic diseases, because some of them are treatable. PKU is treatable. It just means you have to be careful. Phenylketonuria is an inability of breaking down an amino acid called phenylalanine. So all you have to do is just avoid phenylalanine for the rest of your life. And you'll be, the rest of your life could be completely normal. Some of them not like that. There's nothing you can do about it. Well, albinism is not, nothing you can do. Cystic fibrosis, the most common genetic disease in human beings, you can't do anything about it. Sickle cell, you can't do anything about it. Tay-Sachs is really bad. It causes death in childhood. There's nothing you can do. Some are dominant, which means you only need one. Now, remember, the recessive ones, you have to get a small letter from both parents, right? So both parents have to be carriers. Dominant disorders, all you need is one. That's it. One of them from one of your parents, and you got it. So achondroplasia is dwarfism. What's one type of Alzheimer's? Something called Huntington's disease is brain degeneration in your late 40s, early 50s, and hypercholesterolemia, excess cholesterol in blood. These are all dominant disorders. Okay, so most genetic mutations that cause medical disorders are recessive. We saw a few of those were dominant, mostly recessive. Fortunately, individuals who have the recessive allele, but appeal normal, are called carriers of the disorder. They can pass it on to their offspring. Cystic fibrosis is the most common lethal genetic disease in the United States. Well, 1 in 31 Americans, or 10.5 million Americans, are carriers. It's not immediately lethal. People can usually live into 20s to 30s or so, but then they usually don't live, so they have very short lifespans. Using Mendel's laws, we can predict the fraction of effective individuals that likely result from a marriage between two carriers. So there it is. So here is albinism, a recessive disorder. Normal pigmentation in two parents, but they're heterozygous. What is the probability that they'll have an albino child? 1/4. Because you just do it on a square. Big A, small a for the sperm, big A, small a for the eggs. 1/4 of them will be big A. 1/2 of the the prediction is that 1/4 of the children will be big A. 1/2 of the children will be big A, small a, and 1/4 will likely be albino. So if a man and woman who are both carriers of cystic fibrosis have 3 children without cystic fibrosis, if a couple has a 4th child, what's the probability the child will have the disorder? Still 1/4. Every child, the probability is 1/4. Doesn't matter how many child you have earlier that don't have the disorder, each one is going to be 1/4. Right? It's still 1/4. 25% chance. A number of human disorders are caused by dominant alleles. Example is achondroplasia, from a dwarfism, with a head and torso developed, but the arms and legs are short. So the homozygous dominant genotype causes death of the embryo, and therefore only heterozygotes survive. So this means that if this person with this dwarfism has a child, there's a 50% chance that their child's going to be a dwarf also, because they got a big A and a small a. If they give them a big A, dwarfism. It can be a small a, not a dwarfism. Do you see that? Okay. So there it is. And this is like the test course I showed you before. So here's the sperm. So dad is normal. Mom is a dwarf and has a big D, so half of the children are going to be heterozygous, and therefore, the probability is that half of those children will be dwarfs and half will be normal. So dominant alleles that cause lethal disorders are much less common than lethal recessive alleles, fortunately. Huntington's disease, the degeneration of the nervous system, in middle age is one of them. And once it begins, it's irreversible and inevitably fatal. And since it's dominant, any child born to a parent with the allele has a 50% chance of passing it on. And if you know you've got it, there's nothing you can do about it. You know that your brain is going to degenerate in your late 40s, early 50s. So it's really a bad one. Okay, I'm going to stop right here. And so this one will be 106 genetics 2. So catch you in the next video.